Hi guys, it's Monica, Microbiology 225, Azusa Pacific University, Professor Karen Stansel. So this week I'm working on a couple of experiments. First experiment has two parts, and the first one that I will be doing is biochemical testing for microbial identification, which involves methyl red, the Voges, Proskaeur, and the Catalase testing. So, excuse me, as you can see, I have all my stuff set up, but I won't be using everything. I'm so sorry, I won't be using everything because it is in two parts and there's something that I have to incubate. So, I've cleaned my area, I've gathered everything that I need, I washed my hands, I have my gloves on, I have my apron. So, we're going to start with labeling my MRVP tubes. So I got two tubes right here. There's 1 and 2. So I have to label that with, get a marker right here. Give me 1 second. Okay, sorry about that guys, I'm back. I've labeled my tubes. One is E. coli and one is the S. epidermis. And I have my active broth cultures here that I will be using. So I'm going to pull out the epidermis and the E. coli. Have them here. We actually don't need these test tubes in this part of the experiment until after the 48 hours after I incubated those. I'm going to take these. I'm just going to line them up with the right name. Okay, so let's get started. So I have my safety goggles on. I labeled that. I have two dropper pipettes in the disposable cup of alcohol. I lit my candle. So I'm going to start with gathering the active E. coli culture, which is this one here. You can see it. I'm going to take this and I'm going to remove the cap and pass the lip of the valve over the flame to sterilize it. And then I'm going to remove the cap of the broth, the MRVP broth one that's labeled E. coli. And I'm going to do the same thing. I'm going to remove one dropper. I'm going to carefully insert the pipette into the active E. coli culture and drop a small amount. So this is the active E. coli. So after doing so, I am going to place the pipette. I'm going to carefully pipette 4 drops of active culture into the MRVP tube. 4. 1, 2, 3, and 4. So I'm done with that. Put this here because I've already used it. Fill that with bleach as soon as I'm done. And then right before I close these up, I'm going to flame them. This is the active E. coli. And this is the MRVP broth, which now has active E. coli 4 drops. And I'm basically going to do the same thing for the epidermis. Take the epidermis. I'm going to flame it. Take this, the MRVP. I'm going to flame that. Take a new pipette. Get a couple drops of epidermis. And the same thing, 4 drops. 1, 2, 3, and 4. So now that that's done, the next step is to place the MRVP cultures into my incubation location, which is right up there, as soon as I get everything cleaned up. And I'm going to incubate that for 48 hours. And then with my cultures, my active cultures that I have here, I'm just going to put them back and use them for future labs and just keep them stored. So once this has incubated for 40 hours, I'm going to check it, and then we'll do part 2, which is the methyl red test. See you in 48 hours. Hi guys, this is Monica again with Microbiology 225, Azusa Pacific University with Professor K. Stancil. So we're now going to be working on part 2 of the biochemical testing for microbial identification, which involves the methyl red, Voges, Proskaeur, and the Catalase testing. So after incubating both of my broths, this is epidermis and this is E. coli. I'm going to move on to the second part, which is pouring the E. coli into both of these test tubes, and I have to equally divide that. So let's see if I can get this. Carefully. So this is E. coli. So this is pretty equal. And now I will do the same with epidermis. So what we're going to be doing after pouring these 8 different reagents, but we're basically looking for any growth. So now we're going to move on to the methyl red reagent tester. So I'm going to take the methyl red, which is this one here, and snap it open. Basically what I'm going to do is I'm going to pipe it 7 drops of methyl red reagent into both E. coli and both epidermis. Actually into one of the E. coli and one of the epidermis. So 7. We'll start with this one. 1, 2, 3, 4, 5, 6, 7. I'll do it into this one. 1, 2, 3, 4, 5, 6, and 7. So we're immediately observing for a color change. So the color of the broth after adding the drops should change color. If the test is positive, the broth will appear red or pink in color. So we can see that there was growth in both of them. Okay, so I'm going to take a picture of this. I'll be back. Guys, so now I'm working on the last part of the biochemical testing for microbial identification. So now we're going to do the Catalase testing, which is actually the last one. So basically what I'm going to do is I've already set up my lab. I have my 2 slides. One is E. coli, one is epidermis. I've drawn my circles. I have my plates, my E. coli and my epidermis. So with my inoculation loop, I'm going to take it and I'm going to remove the lid of the E. coli culture plate, and I'm going to collect several colonies with the inoculation loop. And I'm going to transfer the collected colonies to the circle on this E. coli slide. And after this, I'm going to pipe it 1 drop of hydrogen peroxide. See, so the whole reason for this test is to observe if the formation of oxygen bubbles. When we take a closer look, there's bubbles forming. So that gives me a positive result for E. coli. I'll leave this here since I'm going to use it again. So I'm going to do the same thing for the epidermis. I have my new loop. It's already been cleaned. This is the epidermis. Take a few colonies. And let's see what happens. So actually both form bubbles. It looks like the epidermis created more bubbles than the E. coli. And that concludes our Catalase testing.